All right, hello guys. So today I'm gonna be covering the molecular biology of the cell. It's a lecture that you had. I'm just gonna go over it again with the notes that I have, basically. Okay, so like what we will be covering today is transcription and translation. Principles of regulation of gene expression. You know what basically, and how does a cell decide what gene to transcribe? We're gonna talk about cystic fibrosis and sickle cell anemia, and then briefly we're gonna talk about how some mutations lead to dominance or dominant inheritance patterns, and how some other mutations can lead to recessive inheritance patterns. And then finally, we're gonna cover somatic and inherited mutations. What's the difference between them? And we're gonna have Huntington's as an example at the end. All right. So now we're gonna first look at transcription and translation. This is where like the bulk of the presentation is about. So we're starting with, let's see. First of all, let's differentiate between RNA and DNA. So RNA structure, a single helix containing ribose sugar. Its function is basically a genetic message that is outside the nucleus. It's the expression of the DNA outside the nucleus, basically. The nitrogen bases, of course, you know, in RNA, you have uracil in the place of thymine. So you have adenine, uracil, cytosine, guanine, while in DNA, it's more of adenine, thymine, cytosine, guanine. The location of the RNA is outside the nucleus mainly because it needs to be translated to proteins in ribosomes and rough endoplasmic reticulum, while the location of DNA is inside the nucleus, and it's getting replicated inside the nucleus. It gets transcribed into RNA inside the nucleus as well. Now, function of DNA, you know about it. It's basically that it tells your cell what to do, basically. So it contains the code, which from that you can do several things to the cell. And the structure of it is double helix structure. And to differentiate, of course, RNA and DNA are both nucleic acids. RNA has the ribose sugar, while DNA has deoxyribose sugar. That's like a main difference between them as well. Now about sugar differences, I mean, I just wanted to, I put this in just to like show you the difference where basically. So in the ribose, you have two hydroxyl groups here. In the deoxyribose, only one. The other one is a hydrogen atom. In uracil and in thymine, the differences between them, in uracil, one of the carbons, instead of having the methyl group, you know, organic chemistry, methane methyl group, CH3, instead of having that, in uracil, you only have one hydrogen atom in that place. I mean, it's not a detail that you need to be aware of that, maybe because they might ask you about, they can ask about several things. It's open play basically here. So, you know, just be aware of that. Okay. Now we're moving to like the central dogma of molecular biology. So it's basically a rule that the flow of information, I mean, if you take genetic code as an information, as a source of information, the flow of it is from DNA to RNA and to protein. A small exception is the viruses, some viruses that use reverse transcriptase, which basically makes their RNA, some RNA viruses have RNA, they enter the cell, other than using the cell's machinery to transcribe its own protein, to translate its own proteins and make its own proteins, it integrates its genetic information into the DNA of the host by reverse transcriptase and this stuff. So that's a small exception, but in general, you have DNA to RNA to protein. Now, types of RNA, I'm going to talk about 5 types. And I mean, I don't think you need to know anything more than those types. You have the messenger RNA. It is what's basically being transcribed. You have the RNA that you get after transcription is the mRNA, or to be more accurate, pre-mRNA. We will get to that later. Transfer RNA is the type of RNA that basically brings the amino acids to the codon in the ribosome by connecting to the codon of the mRNA by its anticodon, by the tRNA's anticodon. So it basically brings the amino acid to the ribosome so that you can get the growing peptide chain. The ribosomal RNA, you see, ribosomes are considered as enzymes. They make the protein, and a part of the ribosome is made of protein, but the other part is also made of ribosomal RNA, rRNA. So it's a mixture of both. Heterogeneous nuclear RNA, or more simply pre-mRNA, is let's say the fresh mRNA. When it gets transcribed unedited, that's the mRNA that is called pre-mRNA, because of course you have to have some post-transcriptional modifications on the pre-mRNA we're going to talk about later. MicroRNA, this type of RNA has a role in regulating what genes get, let's say, translated or not, what RNA gets translated or not. It basically inhibits some RNAs from being translated. Okay. Now, let's talk about transcription. And you're going to find that with transcription and translation is that you have, first of all, initiation, elongation, and termination. Those are like the 3 stages of transcription and translation. And of course, they differ. So first of all, in initiation and transcription, you get DNA molecule or a DNA strand right here. You have promoters and specific transcription factors that basically it choose this DNA strand to be copied, basically, or to be translated. And you have the promoter region where transcription starts. And this is where like all the transcription factors and the RNA polymerase 2 as well, to be specific to, in eukaryotes, RNA polymerase 2 is what transcribes our genes and our DNA, mostly anyway. And this is where the promoter region is where the transcription factors, the RNA polymerase 2, other promoters as well, hook onto the DNA and start the transcription process. Now, elongation. Elongation is basically, as you have it in DNA replication, you're going to have it here as well. It's kind of the same process, but only with adenine and thymine, you get only adenine and uracil, not thymine. So that's like the difference. And also, you're only copying 1 strand, not the 2 strands. So what happens here? You have basically the DNA is divided into 2 strands: the coding strand and the template strand. You see, the coding strand is the strand that doesn't get doesn't get transcribed, but it has the same code, the same sequence as the mRNA, of course, accounting to the uracil thymine contradictions, but it basically have the same code of the mRNA that's getting transcribed, the coding strand. While the template strand is the strand that gets transcribed, and it has the opposite sequence to the RNA because it's getting transcribed from there. A note, to note is that, of course, the RNA polymerase synthesizes RNA from the 5' to 3' direction. And so if the RNA polymerase synthesizes from the 5' to 3' direction, that means that the template strand, or the strand that's getting transcribed, is opposite to it, which means the template strand runs from the 3' to 5' direction. And to build on that, the template strand, the strand that is not, the coding strand, the strand that is not getting transcribed, has the same direction, 5' to 3', as the RNA that's getting transcribed. So you have the coding strand doesn't get transcribed, the same sequence and direction as the RNA that's getting transcribed. The template strand that is used for the transcription process gets is the strand that's getting transcribed. It has the opposite orientation and the opposite sequence to the RNA strand that's getting transcribed. All right. Now, termination, the transcription factors, basically the transcription complex consisting of the RNA polymerase transcription factors, various other proteins, it goes through the DNA, until it reaches the termination signal. The termination signals vary. There are a lot of termination signals, but what you need to know is that when it reaches the termination signal, the RNA that is being transcribed dissociates. The RNA polymerase and the complex also, the transcription complex also dissociates, and leaving the DNA back to where it was. So this is where termination finishes, at the termination signal. Okay. Now after transcription, what do we have? We have the pre-mRNA strand. Now, the pre-mRNA strand is basically what you get after you do transcription, and it needs some editing before it can go out, because remember, we're still in the nucleus now. We need some editing before it goes out to the cytoplasm and to the cell, where it gets transcribed, translated, in ribosomes, in the rough endoplasmic reticulum, or free ribosomes as well. So what happens is the primary RNA transcript gets edited. How does it get edited? Well, first of all, you have the addition of a 5' cap at the 5' end of the DNA, of the RNA, and the addition of a poly A tail at the 3' end. Poly A tails means like basically a lot of A's, a lot of adenines. Poly A tail, the 3'. And at the 5' we have a cap. Now, what does this do? Now, the 5' cap serves for the basically helps the ribosomes to attach to the mRNA. It's like a ribosome binding site, and it also protects the mRNA from degradation and from it being digested like a lysosome or something like that. Now, the poly A tail also protects the message against rapid degradation, and it aids in its transport throughout the cytoplasm. Okay, so you have the addition of the 5' end and a 5' cap at the 5' end, and at the 3' end you have the poly A tail. Now the third and last, basically post-transcriptional change is splicing, intron splicing, or RNA splicing. So this intron splicing is done by a spliciosome, which is an RNA protein complex, and what it does is that it cuts out the introns, which are a non-coding parts of the RNA, that they don't code for anything, not for proteins, not for anything. They cut those out and attach the exons The exons are like the areas of the RNA that actually do stand for like some genetic, for some codes for proteins. So how this happens is you have the spliceosome cutting out the introns and then sticking the exons together to get to finally get this mature mRNA. Now we used to call it pre-mRNA. Now it's mature mRNA because now it's finished. It's ready for transcription. All right. A quick note before we move on is alternative splicing. Now, alternative splicing is basically, you have your exons. Rather than, doing it like the first, second, third, and fourth exon, you can mix it up and get different proteins which have basically relatively the same function. They're relatively similar, but different actions of the proteins. An example, and a great example about that is, if you talk about in immunology, B-cell receptors. So B-cell receptors are basically have the same structure of the antibodies that get released by the B-cell itself when it gets stimulated, when it recognizes an antigen, so on and so forth. So what makes the B-cell receptor is a sequence of exons of the same mRNA, and what makes the antibody released when it becomes a plasma cell and starts releasing antibodies has a different arrangement of the exons, but of the same mRNA sequence. So that's like something interesting to think about. Now we're going to talk about translation. First, before we talk about translation, we're going to think about the genetic code and how does it work? How does it work? So basically, what you have here is the language of this mRNA is the codons that you have, the three group of letters, 33333, are what basically give you information for what protein you use, or what amino acid you use. So what happens is, like, during translation, a cell, or more specifically a ribosome, reads the mRNA, the message from the mRNA, and you have specific tRNA molecules that have the specific amino acid attach and give off their amino acid. And we're going to look at that afterwards. Now, an important note is that codons are read from the 5' to 3' direction, always. And like what that gives you is like the three sequences, the three-letter sequence of each amino acid, so on and so forth. Of course, you have the 20 amino acids. Each one of them has the specific letters that go with it. Of course, you don't need to memorize that, but just for interpretation. So let's talk about 4 codons that you might need to keep in mind. Is the start codon AUG, methionine, or the stop/nonsense codons at the end, UAA, UAG, UGA. So when you start, you have the AUG, the start codon, always, and when you finish, you hit when termination happens. Termination happens because you hit either a UAA codon, a UAG, or a UGA codon. Of course, those are the most popular. There might, those are the most thought to be the stop codons. Now let's go to what we need for what we need for translation. The basic components are mRNA that you have from transcription, the tRNA that's going to bring you those amino acids, and the ribosomes that are going to catalyze and first of all read and also catalyze the reactions between the peptides to form this growing peptide chain. All right, so now, so we talked about tRNA are molecular bridges that bridge between the amino acid and the codon that codes for this amino acid. And the connection happens because the tRNA has the anticodon of this codon, and they just click together. And also for ribosomes, you obviously have two subunits of the ribosome, the large subunit and the small subunit. Now, the large ribosomal subunit is what catalyzes this reaction and makes your protein, peptide chain, makes this growing peptide chain, and links those amino acids together. The small ribosomal unit has this more intricate function of reading the mRNA and then basically overseeing the accuracy of the base pairing and of like when the tRNA binds to the mRNA with anticodon-codon interaction. It's responsible for this, basically, for overseeing the accuracy of this, so you don't want to get any mutations here, and also for reading the mRNA. More about ribosomes. You have, of course, 3 sites in the ribosome: the A site, P site, and E site. The A site, the A site is the point of entry for the amino acid tRNA. Now, why is it named amino acid? Because the tRNA carrying the amino acid here, it is still an amino acid. It's not a peptide. It's not linked to this peptide chain. That's why it's called amino acid tRNA. The first entry is where the amino acid tRNA enters. You get then the next station or the next site, the P site, is where it becomes a peptidyl tRNA, because the amino acid associated with this tRNA is in this growing peptide chain now. And then the E site, the exit site, where the tRNA exits, and you get and where it finds also its amino acids so that it can go back in the ribosome. So those are the 3 sites, and like I just wanted to mention the amino acid and the peptidyl tRNA just to like give you a clue of why it is APE, not ABC, for example, or any other acronym. So an interesting note: amino acid tRNA synthetases. Now, what would you say is like, okay, it's cool. The tRNA is getting you the amino acid, but how does the tRNA know what amino acid it has? Or how does the tRNA bring a specific amino acid, Because you have, of course, the difference. You have the tRNA molecule and the amino acid. How do they both become paired and they go to the ribosome? How does that happen? Well, it happens through the action of amino acid tRNA synthetases. And how that happens is that you get the amino acid and you get the tRNA that is specific to it. And what happens here is through a catalyzed reaction, you get the binding of those two, of course, with the usage of ATP. And what's responsible for this binding is the amino acid tRNA synthetases. And on the slide, you find the details of how they're connected, the 3' end of the tRNA with the carboxyl group of the amino acid. And the reaction uses ATP for energy. Okay, now let's talk about translation. So the step step one, the first step of translation is initiation. And in this stage, the ribosome gets together with the mRNA and the first tRNA so that translation can begin. And this is something to remember. The only tRNA that doesn't have to start at the A site and starts directly at the P site is the RNA encoding for methionine or encoding for the start codon. This, so what happens? This tRNA sticks to the P site, to the middle of the small ribosomal unit, the P site, and the small ribosomal unit and the tRNA complex move through the mRNA strand that they have from the 5' end until they reach the AUG codon. Once they reach, as you can see here, they're not reaching, they're not. As they reach, they click together. You have the first amino acid, methionine, starting at the P site. And just to note, they walk from the 5' to 3' direction as well, because that's how you read the codons. Elongation now. Elongation, what happens here is that you have the tRNAs, other tRNAs, and we're finished with methionine. Other tRNAs come and they start binding to their respective codons in the A site, and then you have this growing peptide chain coming along. So the steps of elongation are, first of all, recognition. When the tRNA recognizes, when the anticodon on the tRNA recognizes the codon at the mRNA at the A site, then you have the formation of the peptide bond between the protein, the growing protein on the P site, because the protein is growing away from the P site, as we're going to see here. There's going to be a peptide bond between this and the amino acid coming from the A site. Once this happens, you'll have translocation, which the tRNA that was on the A site now gets shifted to the P site, and the tRNA on the P site goes to the E site and then goes out from the ribosomal complex. Now, as you can see in this picture, of course, you have the amino acid coming here. You have here a recognition. It recognizes this three nucleotide sequence with its anticodons right here. Then a peptide bond happens between, or occurs between the amino acid at the A site and the amino acid at the P site. Now, let's just assume this is a growing peptide chain, not just one protein, but it's one protein for simplicity. So this amino acid right here in the A site connects to the growing peptide chain at the P site. What happens then is that you get the connection, and then you get the connection, and you get then translocation, where you have the tRNA that's basically had the previous protein right here, gets now ejected from the E site, and now the tRNA that you had on the A site becomes in the P site. So it's like a factory-ish type when you have steps. Each one, if a tRNA happens to be in the A site, then you have another tRNA exiting the E site. You know, that type of machinery-ish work. So now let's look at the final step of translation, which is termination. Now, in this stage, the finished polypeptide is released, and what causes this release is So basically, you have the smaller ribosomal unit reading the mRNA through, and that then it stops at either the codon, the stop codons, which are either UAG, UAA, or UGA. Now, those stop codons are recognized by proteins called release factors, and what those proteins, those release factors, do is that they mess with the ribosome, and rather than, because you're always adding a peptide, another peptide, or other amino acids, rather than that, it adds a water molecule so that it separates the growing peptide chain from the last tRNA that was there. Separates it by adding a water molecule rather than another peptide bond. After that, the new protein is released, and then it goes to the Golgi, and it gets folded in several issues and stuff like that, which we're going to talk about in this presentation. Okay, we just finished transcription and translation. Shall we have a 2-3 minute break? Yes.
If anyone has any questions, you can unmute your mic and ask, or you can type it down the chat if you don't want to speak. How are you feeling, Khalid?
Yeah, I'm doing good.
You need a glass of water?
Yeah, I just didn't fill my glass of water now, so I'm in a bit of a stitch. Okay.
It's quite a heavy topic, I think.
Yeah, with the transcription. I mean, when I was doing the presentation, I didn't, because transcription and translation took a lot of the bulk of the presentation.
Yeah, even though it just makes it's hard to understand, it just comes up in exams, so I guess useful.
All right. Okay. Let's continue now.
Yeah.
Since there are no questions, let's continue. Now, okay, we're gonna talk about, now the learning objectives are gonna be more brief, not 20 slides for one learning objective. So be patient with us. So now we're gonna talk about regulation of gene expression, major principles of regulation of gene expression. First, we're gonna talk about transcriptional control. What's transcriptional control? Is basically the control on how often a gene sequence is copied into RNA, and then so that it can be translated and so on and so forth. So transcriptional control basically is when you choose what DNA is copied into RNA. That's basically what it is. And it happens through structural chemical modifications, because remember, the DNA is tightly coiled around histone proteins, histone optimers, so that you need, first of all, to unwind that by the acetylation of histones and demethylation of CpG nucleotides, which basically opens up the DNA from being like this to being like this, and you can transcribe what you want. So those are the structural chemical modifications to access any DNA you want. It's not very specific, but when you talk about DNA binding proteins, which are the specific proteins that choose, I want this strand, I want this gene to be transcribed, I want that gene, I want this gene. This intricate control is done by DNA binding protein, also known as transcription factors. They modulate the gene expression and turn it on or off when you want to transcribe something or you don't want to transcribe something. That's it, basically. Now, to talk about post-transcriptional regulation, we already talked about alternative RNA splicing, which you can choose what exons you're gonna attach together and what exons you're gonna get rid of and not really translate after transcribing. And also you have and this is important, the initiation of translation of selected mRNAs can be blocked by regulatory proteins that regulatory proteins that bind to sequences or structures of the mRNA. So what does that mean? That means that you have specific molecules or proteins that can, first of all, either just bind to the AUG or the start of the mRNA and block its translation, or it could bind to the ribosome, cause changes in the ribosome to basically not allow it to translate the genes or the mRNA. So it's also very variable in this situation as well. But what do you want to take home from this is that you have here you have DNA binding proteins or transcription factors that decide what gene is getting transcribed, and here after transcription you have some regulatory proteins that either inhibit or allow the mRNA, to get translated. All right. A short thing about microRNAs. So microRNAs are small single-stranded RNA molecules that bind to the mRNA. And when they bind to the mRNA, they either cause the mRNA to get degraded or they block its translation. So basically, it's like a post-transcriptional modification on mRNAs. And so microRNAs are actually synthesized in a double-strand manner, and then they get diced by the dicer right here. And then you have the separation of the two strands, and the acting strand that you need is gonna lock into the mRNA that you don't want to be translated, and it causes degradation or blockage of this mRNA. All right, now we're going to go through sickle cell anemia and cystic fibrosis quite briefly as well. So sickle cell anemia caused by a point mutation in the hemoglobin B chain. You have in hemoglobin the A chains and the B chains, the alpha beta. The beta chain is the chain that's affected. And what's the mutation is like only one amino acid is changed in the entire sequence of the protein. And as you can see here, the glutamine amino acid in the normal subtype gets changed to a valine amino acid in the affected subtype. And this is what causes the mutation. And what this does causes the protein to clump up in the cells, causes the sickle-like shape of the red blood cells, and it causes distortion of shape of the red blood cells and makes them weaker, and also makes them more susceptible to clot as well. Now let's move on to cystic fibrosis. Another, just also to keep in mind, sickle cell anemia, cystic fibrosis are both autosomal recessive, so that you need two alleles to be present for this for the condition to occur. Now, in cystic fibrosis, what is damaged is the CFTR channel, is what is damaged. And this CFTR channel is responsible for the efflux of chloride from the cells. And what it's also responsible for is to regulate another channel called an ENaC channel. But you're going to get to that in the respiratory block, not now. But what you need to know for now is that it is responsible for chloride efflux across the cell membrane. So if this gets defected, there is no chloride efflux, and you will have buildup of mucus on in the lumen of the cell, or on the apical membrane, luminal membrane of the cells. And this place, mucus is basically, first of all, it disrupts the actions of the cell, And second of all, it's a great microenvironment for bacteria to live in. Which this is why, like with people with cystic fibrosis, I mean, you have like with their lungs, what goes on is like with bacteria live there, and because this specific function is defected, this is what causes the buildup of mucus and also buildup of bacteria within the mucus. Now, what happens here is that a 3 base pair deletion called delta F508, basically the deletion of phenylalanine on the 508th position in the nucleotide, as you can see right here. In the normal person, you have phenylalanine here in the 508th position, but in the deletion of this 508th amino acid, you get the CFTR mutation. Okay, so now we're going to talk about why some mutations lead to dominant inheritance while others lead to recessive inheritance patterns. And it is like simple but not very accurate. What's basically the specific the rule is that when a mutation activates a new function or alternates a structure, it's likely to be more to be dominant. But if it inhibits a function, it's likely to be recessive. And you have examples in Huntington's and CFTR. That was basically it for this learning objective. Now the final learning objective, talking about inherited and somatic mutations. So somatic mutations that can occur from the first cleavage or the first division of the zygote when the sperm and the egg meet, until when the person is an adult, when it goes through adulthood and stuff like that. So it's basically open game. It's basically every single cell in your body except 2 types, or basically a single type of cells, germ cells. Somatic mutations can occur in all cells in the body except for the germ cells, because that's reserved for inherited mutations. In inherited mutations, you get mutations in the sperm and the egg. So in somatic mutations, these mutations can sometimes lead to cancer, or sometimes they don't lead to cancer, depending on the environment, on what's causing that, and stuff like that as well. Inherited mutations, as I said, on the other hand, are germline mutations because they are present in the eggs or sperm. They're not present in any of the other cells except for those. And the main difference: somatic mutations cannot be inherited, but germline mutations or inherited mutations can be inherited. That's like the main thing to take from that. And finally, we're going to talk about Huntington's disease. Huntington's disease is a neurodegenerative genetic disorder which affects your coordination of muscle movement, and it can also lead to cognitive decline and dementia. And what's the type of this mutation? The type is a CAG, or polyglutamine repeat expansion. You see, this is a CAG. It's only supposed to be 1. But you get those repeated trinucleotide addings, and you get this expansion, which basically creates a new protein, or it creates a new function, basically, for that protein. And this is Huntington's disease is an autosomal dominant disease. Okay. All right, guys. Thank you. This is the end of the presentation. Any questions?
If anyone has any questions for Khalid, they can unmute your mic and say something. I'm just gonna drop a feedback form for you guys to fill, and I'll post it on the Facebook group as well. And then, yeah, once you guys have finished, I'll just take the data and collect it. Thank you, guys. Kala, we can just talk after everyone leaves if that's
Okay. Yes.